So I'd like to bring back Heather Moss, who will be speaking on biomarkers of optic neuropathy in adults. Thank you, Heather.
Great, thanks Shannon. I'm happy to be back, and I'd really like to thank the patients who gave their perspective, which is very inspiring to us both as physicians and researchers. So in the second talk today, what I'd like to talk about is some of the approaches we take for measuring optic nerve structure and also optic nerve function. You just heard a lovely overview from Joyce of some of the advanced things that she's piloting to study optic extrusion more. And later on in this session, you'll hear from Alf Dubra and Tony Norsha about some other state-of-the-art research approaches. But what I wanted to talk to you today was about some of the things that are clinically common and also fairly common in research that we may be able to leverage. So one of the ways to think about measuring optic neuropathies, or really measuring anything in general, is that we can measure the underlying cause. So in this case, we have the bright pink drusen here. And so we can measure the size of that, we can measure the location of that. We could measure the structural abnormality. So in this case, we could measure something to do with the optic nerve head, the volume, or perhaps some of the autofluorescence qualities. Or most relevant to the outcome that our patients are concerned about, we can measure the visual function. And I'll go through each of these and talk about them, particularly the structure and function. So we've talked a lot about OCT today, and essentially OCT has really revolutionized clinical ophthalmology. We're able to use this device here. I'm not supporting this particular manufacturer, it's just an example, but essentially you could put your head in like a slit lamp, and it takes a picture in a matter of seconds of the back of the eye. And what we get, instead of this image that we see in the clinic, we get a cross-sectional image. And it has a depth of about 1 to 2 millimeters, meaning we can see the entire depth of the retina and into some of the vasculature, the choroid behind. And we can see the individual layers, and we get resolutions under 10 microns. And with each new year and each new technology, it gets better and better. And what we can do with this is that we can assess the microscopic detail of the back of the retina in living people and also in living animals. So for comparison, at the bottom here are histology pictures taken with slicing through the optic nerve and with a microscope of the optic nerve and of the fovea, and you can see how the layers are very similarly seen on the OCT scan. And in particular, we can see the retinal ganglion cell axons, meaning the cables, as well as the cell bodies, or sort of the brain of the cell, as they come together on the retina and then head back to the brain through the optic nerve. So one of the obvious things that we can use, and you've seen some beautiful pictures of this today, is that we can use OCT to detect and measure structural abnormalities of the optic nerve head. So here's the eyeball on the left, and then the optic nerve head is this structure here, which the fibers of the ganglion cells, fibers from the retina, coalesce and head back to the brain along with some of the blood vessels. So some of the things we can see on OCT scans are the cup of the optic nerve, which is one of the telltale signs of glaucoma when it gets enlarged. With papilledema, which I talked about earlier, we can see things like these little tiny folds here. We can see the elevation of the optic nerve. We can see that there's some deformation of the back of the eye, almost being flattened and pushed into the eye. And then with optic disc drusen, we can see the drusen themselves. And we can also see the resulting elevation of the optic nerve. Some people have been really creative with this and have done things like image the eye looking in different directions. So image it looking right versus imaging looking left versus straight ahead. And you can start to see the mechanical effects of the optic nerve being pulled a little bit as the eye looks around. And that can also give us some information about what structurally is going on. So all of those things that I just talked about can also be measured by processing the images. That's usually done on a research basis. But what we do every day in clinic are built-in routines in the machine that allow us to measure the optic nerve injury. So basically, here's a picture of the fovea. And as I mentioned, these top two layers here are the ones that the ganglion cells contribute to. So the nerve fiber layer, which is the axon tubes, and then the cell bodies. And when you take a stack of these images, as shown here on the right, you can measure the thickness of those layers, and then you can create a heat map. So as an example here, in the right eye, which is shown on the left, the ganglion cell thicknesses are all green, meaning within normal limits, whereas on the left, or sorry, on the left eye, which is on the right of this image, you can see that the ganglion cell layer is thinner, and this corresponds to it being red, and we can interpret that as optic nerve injury. And by using these types of measures, we're able to compare to normative data by different ages from different injuries. We're able to detect change in an individual over time, and then we can also compare the patterns for different insults. So glaucoma versus drusen versus papilledema versus, say, optic neuritis and MS, and that can also help to teach us about the diseases. But a challenge in OCT and other kinds of reflectance imaging, you'll hear about SLO a little bit later, ultrasound also has this problem, is that when you have light coming in, here it is shining at the optic nerve head, it has to penetrate and then be reflected back by the different layers in order to get the images. And basically, in order to get further into the structure, here, you have to use a different frequency, a higher frequency, which corresponds to a longer wavelength. But by doing that, you also lose some resolution. And so there's this trade-off. And that's one of the challenges with OCT. So you can see here with this papilledema, the swollen nerve here, that the back of it is shadowed because the light can't penetrate to image what's back here. You can see in this drusen nerve that's a little less swollen, you penetrate a little bit more, but even in the center here, you can't see back very well. And so it's hard to see what structurally is going on. So one of the things that's useful is some other kinds of imaging technologies. One very familiar to most people is an MRI scan or a CT scan. And I've tried to show the size of the OCT for comparison. So this is a bed that people lie on versus a frame that they just put their head into. And with an MRI, which is much bigger, much more expensive, takes much longer for the patient, we can get these 3D pictures of all the structures around the eye. And particularly here in an eye with papilledema, you can see the cerebral spinal fluid space, which is where the pressure builds up. You can see the structure of the eye, you can see the lens, you can see the muscles that surround the eye. But the trade-off here is resolution. So even with the best MRI, we're getting resolution at about 0.5 millimeter. And 0.5 millimeter is still 500 microns. And remember I said that we're getting measurements on the order of 10 microns with OCT. And so shown approximately in these yellow boxes is the area and the detail that we're able to get in OCT. And so perhaps the real option is to have multimodality images where you're looking at different things together. So now switching gears a little bit to talk about measuring function. So for all of you who have been to have your eye tests done, there's all of these different tests. One of our patients talked very eloquently about how she had this visual field test done, and the doctor initially said, well, I think you just didn't do a very good job, because for any of you who've taken it, it's really what I call to kids the worst video game ever. And we also use measurements of visual acuity and color vision. And a challenge is that all of these systems rely on the patient saying whether they saw something or didn't see something. And this is two problems. Number one, it's completely subjective. And so if there's any struggling with the test or the testing isn't explained well, that can lead to poor results. And it also tests the entire visual pathway. If you don't have your glasses on, if you have a cataract, it will impact the result. If you have an optic neuropathy, it will impact the result. If you have a stroke, it will impact the result. And I don't expect anyone to read this entire chart, but I just wanted to provide a reference that there are some other techniques that can be more objective for measuring ganglion cell function, meaning that they don't rely on the subject response. And I'll talk a little bit about a couple of these. So one option is electroretinography. So the ERG is created when light is flashed at an individual, and then the electrical activity of the eye is measured using this thread electrode that's put in front of the eye. Usually it's done in a very specialized setup in a dark room where there's minimal electrical noise, and you flash multiple times and you average all of these flashes to improve signal to noise, and then this is an example waveform that you get. And in particular, this waveform, the negative waveform after the prominent B wave, is the photopic negative response, which localizes to the ganglion cells. And researchers have worked to optimize what are the flashes of light that best bring out this waveform. And so this is some work that I did in the past looking specifically at papilledema subjects. And you can see that in these waveforms here, that first of all, there's quite a spread. If you look at the normals, which are in gray, compared to the patients with papilledema, which are in colors, you can see there's a bunch that have this abnormal photopic negative response. And indeed, this helps to pick them out as well if you just look at the amplitude. So this is one potential option that could help to give objective measurements. This is one particular patient that I thought had a very interesting result. So her vision with papilledema is shown on the left here. And in her pre-treatment ERG, you can see that the photopic negative response is not very robust compared to the normal in black. With treatment, her vision actually didn't change that much, still showing a mild visual field loss on the right, but the photopic negative response improved. And so this offers the question of whether some of these electrophysiology tests might be able to anticipate vision, ganglion cells that are at risk of losing vision, and with aggressive treatment, one might save, so to speak. Another technique is to use the pupillary light response. So the pupillary light response is very important for controlling the aperture of the eye to help not let too much light in when it's bright and to help let more light in when it's dark. And we can leverage that as well for measurement. So again, by using a similar flash setup, we can record with an infrared camera the pupil size and measure it getting smaller when there is a light flash given. And then we can use the light flash manipulation to change the different color, to change the duration in order to bring out different aspects of the pupillary response. And since the pupillary response goes down the optic nerve to the brainstem, this is often mainly controlled as a measure of optic nerve function, although not absolutely. So one challenge with all of this, and Shannon Barris will speak to this in the future, is that the feasibility of these measurements. So what I've shown you so far was dealing in sort of very controlled ERG labs. The pupillometry setups are using these high, frame rate pupil cameras that require a lot of offline processing, but give us very interesting information. And this is hard to generalize to use in the clinic. And so one of the things we've been working on recently is using some simplified systems, which may not get quite state-of-the-art or quite as high resolution measurements, but might make it more feasible to use in the clinic, to use every visit, to use at the bedside. And this is one such device. By using a skin electrode, you don't have to put the electrode across the eye, although that certainly compromises signal. And by having a handheld system, you can take it to the patient rather than taking the patient to the system. So here's an example of 155 patients we recorded in the top with the ERG and comparing the optic nerve thickness on OCT. So basically a lower number means more optic nerve injury versus the photopic negative response. And remember, a smaller negative number means worse optic nerve function. And although this is statistically significant, with the trend line, you can see by the scatter of the data that there's a lot of issues in terms of noisiness in the signal and perhaps variables we haven't identified. This is actually an issue in the other sort of benchtop ERGs as well, and may in fact be one of the reasons they aren't as in broad spread use for monitoring of optic neuropathies. With the pupil size, we sort of did an aftermarket hack, and this caused a little bit more effect. You see, we were very good at separating the affected eyes from the fellow eyes from the control eyes, and this might be useful in the clinic. So hopefully I've set the conversation there. And then to come will be Shannon Barris talking about some considerations in kids who are harder to study than adults. And then we'll also have Alp Dubra talking about some more advanced structural images, and Dr. Norsha talking about some advanced functional methods. Thank you.
Thank you, Heather. That was terrific. I really enjoyed learning about all those imaging modalities. We'll open up the floor to any questions from our audience. Please type in your questions and we'd be happy to share them with everyone. I'll start off with a question, Heather. On that handheld device that you were testing to look at the pupillary response, do you think that's effective enough to pick up most of the abnormalities? Or do you think it would have so many patients that would be missed that it wouldn't be useful at all to use that?
Right, so I think that's a challenge in the development of any diagnostic technique, is the first thing that we try to show is that we can measure something that's comparable to another gold standard measurement. But then the next question is, how do we do it? If we're trying to detect disease, we want to show that we're able to pick up all the people with disease and maybe a few extras. If we're trying to follow disease, then what we need to do is be able to detect change. And then the real holy grail is to be able to use the measurements to predict the future, to prognosticate what might happen to that patient in the future, and then to have an opportunity to intervene. To be honest, at this point with the electroretinography, the photopic negative response, I think it provides some scientific input, but unfortunately the variability has just been too much, especially with the portable system, to be able to use it to classify people, although we're now starting some efforts with machine learning techniques to see if we can pull more out of the waveform than just the amplitude. The pupillometry does look a little more promising, but because that was more of a development project, the work we've done so far is really just studying it in about 15 patients as a sanity check to see if our protocol works. And now that we've confirmed that it does, we're going to move into testing it more widely in the clinic to see how it performs.
Great. We also have a question from Susan Altman. Her question is, how effective is visual evoked potential testing?
So I'm going to defer that to Dr. Norsha, which is why I didn't tread too much there. But VEP records the potential at the occipital lobe, so at the back of the brain, in response to flashes of light. That is another thing that gets used. I focused more on the ERG because it's specific to the ganglion cells, whereas the visual evoked potential covers the entire visual pathway. Although obviously if you only have a ganglion cell problem, that's what most of the change is going to be from. Dr. Norsh has done some really state-of-the-art things in terms of manipulating the signal and how the light sources are presented to try and get some more scientific insight. So more to come.
Great. Terry Garvey also has a question. Is it accurate to say that the main focus at this phase is on diagnosis and related tools and the need to intervene more than what the intervention would look like?
So I'm someone who's very interested in diagnosis as a research side. I think that both are being pursued very heavily. Obviously, I think for a public health perspective, treatment is extremely important, but you have to remember that you can't test a treatment until you can measure the outcome, and you can't test a treatment until you can make the diagnosis. And so sometimes there may be heterogeneity in the disease itself. So maybe the people with green autofluorescent drusen respond one way and the people with blue autofluorescent drusen respond another way. And until we can divide those out, if we just treat everyone as a group, we might not be able to measure those outcomes. Similarly, if we rely on an insensitive measurement technique, for example, black on white visual acuity, which probably isn't going to be particularly sensitive in measuring vision loss due to optic disc drusen. And so that's not going to be very helpful in studying the treatment. So they really need to come hand in hand to improve patient outcomes.
Great. We have another question from Heather Clem. Her question is, I was diagnosed with optic nerve head drusen when I was 25, and now I am 53. Do you think there's any chance of an effective treatment or cure in my lifetime? And there are any clinical trials?
So I do not believe there's any clinical trials, although again, I think that's a nice preview question to the treatment session that's going to come later today. You know, I think the part of the problem here too is not only treatment, but also predicting progression. So many people in the world don't have drusen. Of the people who have drusens, some of them lose vision and some of them don't. And at this point, we just don't have the knowledge. Maybe we don't have the technology, maybe we don't have the knowledge, maybe we don't have both to predict who those people will be. And the better we can get at identifying who is at risk of vision loss, the better we will be at targeting those treatments and testing if the treatments work. So that's why we're here, to solve that problem.
Wonderful.